Hello everyone, my name is Niamh and I'm one of the researchers in the Fluids and Advanced Manufacturing group working on an interdisciplinary research collaboration tackling hard to treat cancers here at the University of Cambridge. Welcome to this short video on the work we are doing on this project at the Institute for Manufacturing, entitled Imaging Medicine in the Body from Blood and Guts to Brains in a Box. The human body is an extremely complex place, made up of many different cells, which are the basic building block of all human life. They provide structure for the body, take nutrients from our food and convert them to energy, and also carry out specialized functions. Each one of us is made up of 37 trillion cells. To give you an idea of what that looks like, if every one of our cells was the size of a tennis ball, then we would each have enough cells to fill the Royal Albert Hall 1,000 times over. Each one of these cells needs oxygen and nutrients to keep them alive, and this is where our blood vessels and circulation system come into play. Our heart pumps blood all around our body, supplying the much needed oxygen and nutrients to all of these trillions of cells. And this is shown here with a wonderful gift taken from the British Heart Foundation. But what happens when something goes wrong with some of these trillions of cells, particularly if they're in hard-to-reach places? In this case, doctors can inject medicine into the bloodstream, which then carries it all around the body until it eventually reaches any troubled or unhealthy cells. In any kind of flow, particles like the medicine are subject to different forces due to something called shear, which occurs when particles, like the different cells in the blood, slide past each other. This flow and sliding of blood cells past each other is shown beautifully in this animation from a group at Rutgers University in New Jersey, as we can see here. The shear forces are particularly strong when particles have to move from a larger blood vessel into a smaller one, like a capillary. This means that any medicine that we add to our bloodstream needs to be able to bend and squish as it moves between vessels of different sizes. As part of our research, we are building a system to mimic the blood flow so we can investigate how the medicine moves in the blood. And how are we going to do this? Well, first of all, we use a pump to simulate the control that the heart has on our blood flow. And then we're going to use different sized tubings to create the different blood vessels. Here's what our system looks like in real life. We have a pump that leads to different sized tubings that we can circulate over many days. The next thing we want to look at is what happens once the medicine reaches the cells where it needs to react. It needs to now leave the blood flow or leave the blood vessels, and thus its ability to bend and squish is very important once again. The way we investigate medicine leaving the blood vessels is by 3D printing a device that we can add into our blood flow system from the previous slides. This device causes our sample to flow past a material known as a hydrogel, which is used to mimic human tissue in our bodies. Using this device, we can watch the medicine move from the blood flow into the hydrogel in our device. This animation, we can see the blue dye to represent our medicine, and you can see it moving from the blood flow into the hydrogel. The medicines we're interested in particular, are nanoparticles based on polydopamine that are being developed by our wonderful collaborators in the Fruit Group, based in the Department of Chemical Engineering and Biotechnology, also here in Cambridge. Because they're based on polydopamine, which is already found in the body, these particles are biocompatible, and they also have excellent stability, and they have a very low cost to produce. These polydopamine particles are being developed to treat pancreatic cancer, which can be hard to treat because the people suffering from it are not always suitable for surgery, making systemic delivery of nanoparticles through the blood flow a very promising option. So, what happens if the unhealthy cells are not in the pancreas, but this time they're in a person's brain? Things are a bit different in their brain because in the brain there's something surrounding the blood vessels called the blood-brain barrier. This is a layer of cells that protects the brain. It acts like a membrane and prevents any small particles in the blood, which might be toxic, from entering the brain. The problem with this is that it also prevents medicine in the blood entering the brain to treat on any of the unhealthy cells. To overcome this issue, our wonderful colleagues in the bioelectronics group in the Department of Engineering, also here at the University of Cambridge, are developing new methods to deliver medicine to the brain. These devices will use tiny pumps to deliver medicine directly into someone's brain to bypass the blood-brain barrier. Obviously, we can't do experiments with real human brains, so as part of our research, we're building a brain in a box. This is what the experiment looks like. We use a digital camera to watch how drugs are delivered from the device into more of the hydrogel material mentioned earlier that acts as a mimic for human tissue. Our goal with these experiments is to ensure enough of the medicine is coming from the device and is reaching the correct places in our brain in a box. With that, I hope you enjoyed our Frankenstein-style approach to imaging medicine within the body, from blood and guts to brains in a box. Thank you for listening.